What I'd like to do now is take a considerable shift in the conversation, right? We've been talking about things sort of at that biochemical level. We've been talking about ion channels. We've been talking about drugs and neurotransmitters. All of that conversation is still important, right? All of that stuff is still at play here, okay? We just want to move in to a system now, right? So we're going to kind of change our level of focus. Now, while we're thinking about this, we're actually going to still get down to the cellular and the subcellular level, but we're going to be kind of zooming in and out, right, throughout this so we can think about how the visual system works and what you need to know about that. Who remembers that three-step process that I outlined for you at the beginning the other day? It was like input, calculation, and output, right? And I said we can think about this at any level. We can take this all the way down to ion channels. We can take it all the way up to the organism. Okay. Anywhere in between, we can think about this. The visual system is going to be one of those areas in between, right? Primarily, we're going to think about it as an input system because we're going to use it to collect information from the outside world. But there's also going to definitely be some calculation on that. And the system is going to actually send some outputs to other places so that we can continue that calculation process until we make some sort of action. All right, we're definitely going to talk about the eye, the structure of the eye. It's really important to think about the input device so you know what type of information is getting in, what that format is, how that works. We'll talk about visual processing, we'll talk about color vision, form, spatial vision, orientation, movement, all kinds of fun things. We might even watch a movie. It's going to be a good one. It's a Nobel Prize winning video. Well, I should say it's a video from someone who won a Nobel Prize. That's a different story, right? So they won the Nobel Prize and they made the video because they thought they were, important at that point. All right, how many of you are familiar with the eye? If you don't know where it is, do this. You'll find it quickly, I promise. There are two basic processes that we want to think about. One is going to be sensation and the other is perception. How many of you have heard of sensation and perception? How many of you took a whole class on sensation and perception? Yeah, I know, right? You had to listen to me for 16 weeks go on about that, which I'm sure was thrilling. You're not going to have a full 16 weeks this time. It's going to be short. We need to pull these two things apart because they're actually handled in different brain regions. They do different things. They have different things going on. They affect our behavior differently. So we want to think about that. Sensation is really just the cells of your nervous system and the stimulus in the environment. That's it. That interaction between, in this case, we're thinking about vision. So the stimulus in that case is going to be light. So we're thinking about light and how light is interacting in particular with the cells of your retina, which we typically call rods and cones. Okay? That interaction, that's sensation. Beyond that, we actually get into the concept of perception. Now, sometimes the line between sensation and perception is a little fuzzy, right? Definitely we know it's sensation if it's light hitting retina. That's clear. Perception is, what's very clear about perception is that apple looks good. That looks tasty. That's perception, right? The stuff in between, you can shift the line a little bit. But perception is going to be your experience, your interpretation of that information. Okay? That's what we mean by perception. Again, when we think about vision, we're thinking about light. That's our stimulus of interest. Okay? We sort of have three qualities that we might want to describe here. We might think about the hue, right? So what do we mean by hue? We're thinking about the color, right? You might describe something as blue or red or green. That's going to be hue, and it's related to the dominant wavelength, right? So the wavelength of that light, whatever that is, that's going to be perceived by you as some color, right? But we can measure the wavelength, and you can tell us your perception of the color. Brightness is going to be intensity. Okay. So what's kind of, we're thinking about amplitude in this case, right? It might be the same wavelength, right? Peak to peak distance is going to be the same, but that amplitude is going to be higher. It's going to be more intense, right? Finally, we think about saturation or purity. If you think about the color red, for example, the dominant wavelength is obviously going to be what we perceive as red. If you think about the color pink, the dominant wavelength is going to be the same wavelength. It's still going to be it, but it's going to be saturated with other wavelengths of light, right? They're not going to be the dominant wavelength, but you're going to have other wavelengths in there. So sometimes we would say pink is a desaturated red. So that's pretty awesome, right? Anytime we talk about a sensory system, we have to have sensory receptors. Okay? These are highly I'm going to add that here. Highly specialized neurons. I guess all neurons are highly specialized though, right? So maybe I don't need that anymore. Maybe I don't even need that. These are neurons that are built to detect a particular type of external stimulus, right? If we're thinking about vision, again, that stimulus is light. So we have receptors, we have cells, again, rods and cones. We'll talk about those in a minute. That are specifically for the detection of light. Once we talk about the auditory system, we're going to have inner hair cells. Inner hair cells are specific for sound waves. That's it, right? Their structure is different, and it is specific for that stimulus. You're not going to use a rod or a cone to detect a physical, a touch, a poke, right? You've got mechanical, we call them mechanoreceptors. You've got mechanoreceptors for that. You're not going to use mechanoreceptors to detect hot or cold. We have thermoreceptors for that. Okay? So we have different receptors for every kind of stimulus. If you can't see it, if you can't feel it, if you can't taste it or touch it, then you don't have a receptor for it. Okay? A great example of this, we're going to stick with light. How many of you love ultraviolet light? Correct answer is no one. That's the one that causes skin cancer. So you typically want to avoid that one, right? How many of you buy sunglasses? Yeah, it should block out UV light. Does it say that on your lens somewhere, or did it just say these will make my head look good? Sometimes you buy those, right? Casey, I just have to buy the ones that fit my head. I have a lot of options. That's how I ended up with those big Ray-Ban things. I broke my last ones. I had these nice Oakley ones that I thought were nice. They even had these like amber colored lenses, which were kind of interesting. And like they were plastic and the frame broke. It's like, nice display. Yeah, I know, right? I'm going to take those lenses out though, because they're polarized, and put them in that Optimus Prime mask. So that's because the lenses are still good. It's just the frame's broken. It's going to be exciting. So UV light. Hey, do you know who can see UV light? We're going to have a conversation about the birds and the bees today. Some birds and some bees can see UV light. We weren't really going to have that conversation until a couple weeks from now. But birds and bees, some birds and bees can actually see UV light. But that's pretty interesting, right? So if you look at a flower, how many of you love flowers? Try to draw. I don't even really know what a flower looks like. It's like a crab claw. Is that a flower? Just go. It's got leaves. This is why I didn't go into botany. You can tell, right? So when you look at that, you go like, okay, that's just a regular flower. If a bee looks at it, it might have these cool stripes because they can see that under UV light. So how did we determine this? People will take images of flowers using UV light, right? And then when you develop those images, you can see interesting patterns on them that if your eyes are sensitive to UV light, you would be able to pick up on that. So that's pretty cool, right? Same thing with ultrasonic, right? Sounds, right? So we can't hear those. That's why we call them ultrasonic, right? Infrasonic is the other way. That's below, right? Infrared, that's the other kind of light too. How many of you love remote controls? Like for your television? Those typically work on infrared light. That's kind of how that works. They originally worked on sound frequencies. Like the first remotes that were ever developed were sound frequencies. Some people said that their dogs were really disrupted by the remotes, right? Because they would play these like high-pitched noises that you can't hear, but that your dog could hear. And then your dog would go crazy when you were trying to, like switch over to Walter Cronkite or whatever was on at that time. It's like Zane's the only person who thought the Walter Cronkite reference was good. The rest of you, look up Walter Cronkite. So Casey, this is when I'd love to make a joke, but I think like not everybody in the class would get it, which is fine. But then I think some of the people would think they got it, but they would think the wrong thing about me if I said it, and so I'm not going to say it. Yeah. I was just going to make a reference to journalistic standards, and Walter Cronkite sort of is the face of high standards in journalism. I think that's true, right, Zane? I mean, when you think Edward R. Murrow is probably another who's in that category, but definitely Walter Cronkite is. If he said it, you could trust it. It's not the case with everybody.
Not Brian Williams.
Who?
Not Brian Williams.
Yeah, I mean, boy, he got in a little bit of trouble, didn't he? Well, there you go. But Walter Cronkite, as far as we know, that guy always told us the truth. And he's dead now, so I don't think anybody's really looking into it. After he died?
Yeah.
Yeah. We've got to be able to detect that stimulus, right, Alex? We've got to detect it first. Then we have to do something called sensory transduction. Now, this is the same basic process no matter where you are, right? If it's your ear, if it's your eye, your nose, it doesn't matter. We're going to do sensory transduction. Hey, who remembers that guy, Johann Mueller, who said, like, man, stuff's an electrical impulse, right? We have to convert whatever that is, light, sound, an anvil falling on your foot, whatever it is, because those things happen. I saw that in a Wile E. Coyote cartoon. Whatever it is, you have to convert that to an electrical signal. If you can't, Calvin, convert that to an electrical signal, guess what happens? You don't know, right? Because your nervous system runs on electrical signals. And so you have to convert that into an electrical signal. Okay? We typically call that a graded or receptor potential. It's not going to be an action potential, right? But it's going to be kind of this up and down. And again, we can eventually get an action potential. Because this is a sensory receptor, it may not happen. We're going to talk about the visual system today. Your rods and cones do not fire action potentials. They pass that information to somebody else who does that, okay? So they're going to pass that on to somebody else. We'll talk about that. Here's the electromagnetic spectrum. This is really out of scale, by the way, right? This should be like the size of Joan C. Edwards Stadium. And this should be the size of like the coin, like the thickness of the coin they use to flip before the game starts to decide which way you're going to kick, right? Okay, so this is very big, right? We can only see probably, I mean, for our purposes, if you know 400 to 700 nanometers wavelength, that's fine. Now, how big is a nanometer? A nanometer is super, 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 super small. Okay, that's how small it is. Now, the smaller you get, the more energy it carries, right, when you're talking about wavelengths. So out here, we're talking about television and radio broadcast. How many of you ever listened to the radio? How many of you know radios exist? You know that thing that comes up out of the top of a car that looks like a stick? That's called an antenna. And what that antenna does is it receives these, I know, right? These big, big giant waves like the size of a person. Okay? Size of a person. They're huge. They don't really, I hope, they don't really cause much, they don't really carry a lot of energy. If they did carry a lot of energy, guess what would happen to you? Your face would melt off. Okay? Because if we were using, no, seriously, this is true. If we had to use television and radio broadcast waves that were like the size of X-rays, or worse yet, gamma rays, then we would all just turn green and angry. But if we were X-rays, I mean, what happens if you get X-rays? Well, I mean, we can see inside your body. How many times do you get an X-ray? Like as few as possible, right? And the reason for that is these are super small, high energy waves, right? And if you continue to get X-rays and X-rays and X-rays and X-rays, it's going to drastically increase the likelihood you're going to develop like a tumor or cancer, right? So it can really cause some problems. So you want to back off as much as you can. When you go to the dentist, which I hope you all do, like once a year they'll take X-rays, right? They don't want to be blasting your head, you know, every 2 weeks taking an X-ray. That's a bad idea. Okay? This is not to say, like, the next time you go somewhere and, like, you know, you got a bone sticking out somewhere not to get an X-ray. You probably should, right? But don't go in for fun and get an X-ray. This small sliver, that's all we can see. We already said, like, some birds and bees can see things over here. How many of you love snakes? Specifically, who loves pit vipers? Do you know why they call them pit vipers? You can dig a hole and just put a bunch of snakes in there, and then you can throw people in that you don't like. So you put vipers, no, that's not why they're called pit vipers. They're called pit vipers because just back on their heads a little bit before you get to the neck and the tail, which is not that much space on a snake, by the way. When you get back there, they actually have these infrared sensitive areas, right? And so they can sense infrared, which we typically think of as heat, right? So they can actually sort of get a heat map of the world. There's this guy in Germany, and I think he has videos even online. He takes pit vipers, and this is all for science, by the way, Juwan, so just let you know. He'll grab a pit viper, and this will definitely put a pit viper in a great mood, I promise you. He wraps electrical tape around its eyes so it can't see. Yeah. And then he has to put it back in its cage. And the proper technique for that is this. Because you don't want you're afraid it's going to bite you. So you just kind of fling it, I think, is how that works. So he puts the pit viper back in there. And then what he will do is he'll put a rat. Pit vipers eat rats. He'll put a rat in there. Now, how many of you have, and you should do this the next time you get the opportunity, Riley, not to you, but to one of your friends, to wrap their head in electrical tape, right? So if you'll wrap their head in electrical tape, what will happen is they'll not be able to see, right? Because electrical tape is black and it's opaque, so you can't see through it. So the snake can't see, but guess what it can still do? Right onto a rat, because it's got those infrared sensing pits, right? So it can still create like a map of its world as something hot is moving through it like a rat. I have no idea how you remove the electrical tape from the snake. Because I think after I've grabbed it that one, I don't know. I'm sure there's a technique. That's a true story. I met that guy. Yeah.
Oh, is that how you know how to throw them? Did he show you?
Yeah, he did. He did it with a fillet of fish. He had some trout while we were eating dinner together. He showed me exactly how to do it. Yeah. Questions about that? All right, let's think about your eye just a little bit. You think about the eye as that receptive organ, right? Now, we're not going to get into a massive amount of detail. We could talk for a semester about the eye and the structure of the eye and how awesome that is. And it is awesome, by the way. There are some things you do need to know. First is you need to know about the retina. This is... Do you know what's cool about the retina? It's the only neural tissue I can see without cutting your head open. Right? How many of you have been to the eye doctor? Yeah, I can tell which ones of you have. I can tell which ones of you should go to the eye doctor. Those are the ones of you who can't find the door on the way out. So you get your eyes checked. So when you go to the eye doctor, and they do part of this just for fun, but part of it's diagnostic. They get that bright light. Remember that ophthalmoscope that we said that Hermann von Helmholtz created? They shine it in your eye, right? They're looking at your retina, right? And they want to see the structure of your retina. Now, there are certain things they're looking for. There are certain diseases, disorders that cause problems with your retina. Diabetes is one, right? So they'll definitely check your eyes more often if you've been diagnosed with diabetes. Some folks have macular degeneration, right? That's kind of like and that happens to you. There's nothing you're going to do to prevent it. There's age-related macular degeneration, but then there's also sort of advanced, macular degeneration that can happen. Your retina can actually become detached from the back. Yeah, that's the face you make right there, Celine, because that's not fun. What can happen there is when your retina becomes detached, you can actually go blind, right? Because your retina can just fold up and you're not going to be able to detect light anymore. So I'd watch out for that. So walk around gently, right? Don't blink too hard. Or blink more often. That'll just help your cornea. So the retina is the only neural tissue that we can actually see without getting inside your body, right? And so we know a lot about it because there are a lot of people who will line up and say, hey, you can look in my eye. There are not a whole lot of people lining up saying, hey, you can look inside my head, right? So it's a lot less invasive. And so there you go. So there are what we call photoreceptive cells. What do we mean by photo? We're talking about light, right? Again, these are the rods and the cones, right? They're going to be in your retina, so that's awesome. We call those guys photoreceptors. Okay. They take that light energy and turn it into those electrical potentials, right? They're going to collect photons of light. Now we're not going to get into a whole discussion about, because I've sort of switched on you here, and I didn't give you the warning. I talked about light as a wave for a while, and then now I'm talking about it as like photons, right? Like particles. Light can do whatever it wants, so we're just going to let it do that. It can be a wave, it can be a photon, it can be a lot of things. Okay, so we'll talk about it as we need to, however we want to. It's going to collect those photons, these photoreceptors are, and it's going to turn it into that electrical energy, so we can send that information into your brain, right? If we just continued to send it through as photons, guess what would happen? Nothing, right? Because we don't have a way to understand photons inside our brain, right? We can understand electrical impulses, we have to convert. And that, remember that sensory transduction, right? That's that process we're talking about. Now this is a very long process. We're going to get photons of light are going to hit these photopigments. They're going to cause those photopigments to change shapes. They're going to, muscle around on a bunch of other things. We're not going to talk too much about that. But we do have rods and cones. Rods are sensitive to low intensity light. These guys are what you would use at night in a darker environment, right? They're going to get activated with just a little bit of light. Interestingly, most of your rods, well, let's say all, are actually located in what we call the periphery, right? They're not in the central part, what we call the fovea. That's where we keep our cones. These guys require high levels of light. They're also your color receptors, right? We'll talk about color vision later. Okay. Now, why is this interesting? How many of you have ever thought you saw something at night, and then when you looked at it, it disappeared? Okay. Sometimes it's a guy hiding behind a bush who only pops his head out just for a few minutes and then, ducks back down. But most of the time, it's because of the way that your retina is structured, right? So most of the time, we're diurnal animals, right? We're awake during the daylight, so we're going to be up. When we're looking at things, we want it to focus, on the central part of our retina, right? What we call the fovea, okay? So we're going to look at that directly. We're not trying to look off to the side and see things, get a good image that way. At night, though, the problem is your fovea doesn't have any receptors that are sensitive to low levels of light. So if you look directly at something at night, it will disappear, right? Okay? But you can see it out of the corner of your eye, right? You can see it out of the periphery because that's where all your rods are located. So when you see something, you think it's over right, and then your natural process is to look at it directly, and then it's going to disappear because those photoreceptors need more light than what's available. So that's interesting, right? Has that ever tricked you into thinking a ghost was after you? Right, when you were a little kid? Yeah, just go ahead and admit it. I know you were in bed at night and you were, what was that? And you like look and it disappears and you look away and you can't really see what it is because your rods are not great at spatial vision, form vision, things like that. You know, I don't know what that is. So that's kind of cool, right? Hey, what if you were a nocturnal animal? If you lived at night, you'd probably have a different structure, a different setup with your rods and cones, right? Because you would want to be able to see things at night. So that'd be kind of exciting. Hey, look at that. That's an eye. It's got a bunch of muscles around it that move it. You know, you guys can just move your eyes around however you want to. You should all practice that from time to time. Strengthen your eye muscles. Does anybody do that? Nobody does that. They should. I do it all the time. I cross my eyes, see how far I can cross them. It's good for like magic eye diagrams. I'm really great at those. Keep my eye muscles strong. Sometimes you do a little steroid injection in the eye muscle. No. Watch out, I'm going to come in one day, Zane, and my face is going to be bulging right here. So yeah, I just came from the eye gym. Just did 3 sets of 25 eye crosses. Feeling the burn. Hey, Adriana, we're going to add this to our airbag for the brain company. We're going to start an eye muscle regimen.
I just keep imagining like the people at the second floor of the wreck. It's just like just sitting there, crossing their eyes.
Yeah, you know.
Drinking like the NeuroSafe.
NeuroSafe, they got to drink their NeuroSafe, right? Or whatever other garbage they just bought
NeuroSafe.
that they think is going to help them, right? Like, I'm going to eat this elk antler, whatever. Right? No offense to anyone who eats elk antlers. But I don't think they're going to help you. Isn't that a supplement? Anybody know?
It is. I worked at GNC over the summer.
Okay.
It was funny. It was a funny job.
There are like 3 things that'll help you work out. Well, 4 if we're not talking about steroids, because there's that. But basically protein, creatine, and caffeine are the only 3 things that are going to help you out in the gym. So there you go. Yeah. NIH has this nice chart for anybody who's interested in like supplements. And they have like all of the evidence supporting or not supporting particular things. You can look at it. So there you go. Eye crossing. Yeah, there you go, right? Hey, we could sell. Hey, you still have a connection at GNC? What if we could make a specific like eye muscle sort of pill.
Going back to it's my summer job, so I'll go back this summer. You can just give y'all. Like,
We'll have time now
ideas. Yeah.
to get some people hired to make those pills for us.
Yeah.
And then we'll...
Pills in, you need eye drops too. So
Eye
people
drops.
want that, like,
You got to put it in
automatic.
directly. Yeah, that's
They
it.
want- They want that instant.
They do.
Ready to cross.
And you know what? We'll do what they did with toothpaste. We'll put a little citric acid in there so they can feel the burn.
Yeah, absolutely.
That's the reason you brush your teeth. I'm telling you right now is because it had citric acid in it that made you salivate so you could feel it. Prior to like World War I, nobody brushed their teeth. And then the government was like, man, this is like a national security issue because we got all these guys with like rotting teeth trying to fight for us, and it's not really working. They're like, how can we figure this out? And so the Pepsodent marketing guy was like, I don't know, we'll figure out, can you feel the cleanliness? They'd accidentally put in some citric acid in the Pepsodent, which other toothpaste hadn't had up until that point, and that's kind of what made that sort of tingly feel. You know, like when you brush your teeth, you're like, man, I got a good tingle. Nobody says that, but you feel it, right? And that has nothing to do with the cleanliness of your teeth. It's simply an irritant that makes you feel tingly in your mouth, so you like can feel that it was actually working. So there you go.
Learned about that in Psych and Personnel.
Yeah. Yeah. So did he talk about the book Outliers?
Yeah.
He outlines the story in Outliers. It's actually a pretty fascinating story. All right, so here's your eye. Whether or not you've, jacked up your eye muscles, that's up to you. Light is going to come in like this, in through your, it's got to come through the cornea first, right? So that's something you don't want to screw up, okay? So don't take sandpaper to your face. It's a bad idea. So you don't want to scratch your cornea. You don't want to get a corneal ulcer. What are some other things that can happen to your cornea that people get fixed occasionally? Cataracts, yeah. So that's something. You're not going to be able to avoid cataract. Well, you can. I wouldn't recommend it. The only way to avoid cataracts is to not get old. The only way to not get old is to like stop living. Yeah. So, I mean, it's a surefire way, but there you go. You've got your lens. Now, your lens is kind of cool, right? How many of you have ever dissected an eye? Right? Yeah. So that butterscotch that was in there, yeah, that was the lens. So if you've ever cut open probably like a cow eye, is what most folks have done, right? There was like a butterscotch looking thing in there, and hopefully your teacher told you not to eat it because it did look just like a butterscotch. But that's the lens, right? It was kind of hard. Now, amazingly, it bounces.
Yeah.
I haven't tried that. I have to do that.
I got
That happens. You can cut it though and see all the layers that come out. That's what I did, which was a little tricky. But the lens, amazingly, you can actually bend it, right? Okay, and you can bend it with these muscles, right, that are connected to your lens. You need to do that so you can focus on things that are close or far away. Now, again, as you get older, you become less and less able to bend that lens, right? And that's why, I don't know, I remember my mother-in-law in particular, her arms for some reason were not long enough for her to read the menu at dinner, right? And so when you see people of a certain age, right, and maybe you're seeing your parents do this, right, when they go out to dinner and they hold the menu way out there, they're trying to read it, that's when you know they need glasses, and you should mention that to them. And also, don't let them drive. Because, I mean, they can see things far away, but then I don't know what happens when it gets a little closer. I'd be a little worried. So you can kind of notice that. There are also people who have always worn glasses, right? Maybe you're one of those people. That's fine. That happens sometimes because your lens is not the right shape for the size of your eye, right? If you're, if you're nearsighted or farsighted, right, is what we might say there. So what happens there is when your lens tries to focus the light, it needs to focus it right on that retina, right on those photoreceptors. If for some reason the focal point is here, or the focal point is here, guess what you see there at your retina is going to be fuzzy, right? It's going to be the same thing if we ask somebody to climb up on the monitor and start twisting the focus knob, right, or on the projector, you would see things get fuzzy, right? But if we were to move the screen accordingly, you would have a different focal point, right, a different focal depth. So that happens. How do you correct that? You typically correct it by putting another lens in front of the lens you already have, right? That'll do a little pre-bending of the light before it comes in, and then your lens can do whatever it does, and we can get it in there. So that's pretty cool, right? Anybody excited about that? No, nobody's excited about that, right? And if you don't wear glasses now, give it 30 years and you will. It's probably about the time you hit 50, that's when it starts to happen, right? So hang in there, Casey. I know you've always wanted glasses. And these people trust you to be their TA. Hey, while we're talking about that retina, let's talk about some cool stuff. We already mentioned the fovea. This is part of the retina. This is where you have the most what we call acute vision. This is where most of your photoreceptors are jammed in there, right? And it's going to be all those color-sensitive cones, okay? That's all that we're going to find in the fovea, right there. This is why when you look at things, you focus it right kind of in the center, right? You're not going to be looking off to the side to try to... I hope none of you try to read like this, right? That doesn't do any good, okay? It doesn't work that way. Your best vision, central vision, that's where you want that. Now, your retina's in here backwards, right? So we'll show you a picture of the structure of the retina in a minute, but light has to come through the whole retina first, and there are 2 or 3 cell layers it has to go through before it gets to the rods and cones, to that light-sensitive portion, right, Calvin? And then we're going to absorb that photon. Now, it's in there backwards for a reason, because we need to supply nutrients to the rods and cones, and they have to be there at the back of the eye. There's the retinal pigment epithelium, right, that supplies with those nutrients. You don't have to think about that right now, but it's in there backwards. Because it's in there backwards and light has to come in, and then that signal has to come back out, we're going to have a spot where the axons of these cells, remember those axons, they've all got to come through. We got to gather those up and send that back to your brain, right? There's going to be a spot where we punch through that retina, and we're going to call that the optic disc. And this is that exit point of all of those fibers. We can call those axons if you want. Okay. That's eventually going to become the optic nerve. Now this creates something called the blind spot. So there is a spot in both of your retina, right, in both eyes, where you have 0 photoreceptors. If you have 0 photoreceptors, what can you see there? 0, nothing, right? Because you have to have a photoreceptor to see. Now, I believe you can find these tests. Do I have, there it is. So we'll go back a few slides. This is a blind spot test. This is not going to work for you because it's the wrong size and the wrong distance, okay? But you get on a little card, you can get it, you can find these online. They're kind of interesting. If you were to stare at this spot, close one eye, you got to close an eye, and then you were to move this back and forth, and then you get it kind of in this range here, and then that green spot would disappear. Because right here, where the optic nerve comes out of the back of the eye, is a spot where there are not any photoreceptors, okay? And it's kind of interesting, because when you do this, and that green spot will disappear. It's really fascinating. Now, why don't you normally see this? There are a couple things. Jakia, one, your eyes are always moving, right? Even when you're fixating your eyes, they make these little small micro movements, right? So you're trying to like move that blind spot around to different places, right? And so then you can kind of get a nice little, well, here's what's in this area. The other thing is once you get back here, right, and you got the rest of that brain thing going on, your brain will sort of pull a Photoshop, right? And it will just sort of fill in what's there based on what it's seen around it and the other movements you've been making. So that's kind of interesting. All right. So we know we have rods and cones in our retina. What else do we have in the retina? You got these guys called bipolar cells. Okay. These are going to connect the photoreceptors to the ganglion cells. Now what's interesting about the ganglion cells is these are the first guys to fire an action potential. Nobody else in the retina has fired an action potential yet. We had the rods or cones, we had the bipolar cells, then we get into the ganglion cells. These guys will send an action potential through that optic nerve and back into the structures of your brain, okay? So that's simple. In between here, you have a couple other types of cells. So you have what are called horizontal cells. These guys will connect kind of up here with the bipolar cells, back and forth. Amacrine cells are here with the ganglion cells. They do something similar. That doesn't make a whole lot of sense, but we can show you this on this fun diagram, which irritates me to no end. Because someone made a drastic mistake here. And the mistake they made was they bothered to do a couple things. One, they gave you a whole bunch of rods, and really you should have a whole bunch of cones. Two, they put rods and cones mixed together. Rods and cones are not mixed together, okay? All your cones are in the fovea. All your rods are around that. The only place where rods and cones will be adjacent is the transition between the fovea and the periphery, right? So that's going to be the only place that happens. Otherwise, this is an okay diagram. So you've got your photoreceptors. Now this is the eye. Light comes in, goes through all of those. Remember, this guy back here is the retinal pigment epithelium, right? We're supplying nutrients to what we call the outer segment of the rod or the cone. Why do we call them rods and cones? Look at that. It's shaped like a cone. And that's shaped like a rod. It's really just based on the shape, right? That's all we got. So once it comes in, we're going to catch the photons here, turn that into an electrical signal. We've got our horizontal cells in this layer. Okay. They're going to help with some information transfer. We're going to get that into our bipolar cells. We've got our amacrine cells in this layer. And then we get into what we call our ganglion cells. From the ganglion cells, we send that signal back into your brain. Okay. Questions about that? Great. Questions about this? We already talked about that. This is a slightly better, I think, diagram once you understand the way that the light comes in, because we can see here the horizontal cells and the amacrine cells, some of the details of the connections. Again, keep in mind that you would not see rods and cones side by side like that intermingled, okay? But otherwise, this is a pretty good diagram for you to get an idea of how this works. We already talked about transduction, taking light, converting it into a membrane potential, or in our, same thing, electrical impulse. We could think about rods a little bit. They have photopigments in there. I'm not going to ask you much about this, so I'm not going to expect you to get too worked up about the specific photopigments in there. Right. But we should think about how this activity works. This is a concept that we call the dark current. The dark current is that evil impulse that runs inside of all of you that makes you want to just kick old ladies as they're crossing the street. I don't know if any of you have ever done that. No. You've had the urge, though. You got to fight it. I mean, sometimes. What is the dark current really? So let's think about this. Your photoreceptors, not only did we put the entire retina in there backwards, we actually sort of wired up your photoreceptors backwards. Now, the whole story I've been telling you before is that if the cell is activated, it's going to release the neurotransmitter, right? And that's what's going to cause that membrane potential, that current, right? Your photoreceptors are sort of the opposite of that. They will actually, in the absence of stimulation, release neurotransmitter, okay? So they're going to release neurotransmitter when it's dark. Okay? They're going to create what's called the dark current. Now, there's some interesting reasons why we could get into this, why that actually happens. I'm not going to worry you too much on that. The basic idea, though, is when light enters the retina, when it activates the photoreceptor, we get a hyperpolarizing potential, right? Not depolarizing, hyperpolarizing. Normally, this would be depolarizing, right? But in the retina, it's backwards. So we get the hyperpolarizing current. Now, by the time we get to the bipolar cell, because it knows what's happening, it actually converts that into a depolarizing potential, so that when we get to the ganglion cell, we can get action potentials. Okay? If we were to remove this light, then it would be the opposite story, right? Then we would have that depolarizing experience in the photoreceptor, so we would have that current, that dark current. The bipolar cell would then be hyperpolarizing, so it would be negative, not activating your ganglion cell, so we would just have kind of a flat line, no action potentials. Okay. One of the reasons that you might want to think about why this is beneficial to you, when this cell is activated, it stops releasing neurotransmitter. Otherwise, it constantly releases neurotransmitter. Now, if you were to flip this, and you were to constantly be releasing neurotransmitter in the presence of light, you might actually fatigue your cells a bit, right? You might fatigue that. And would you rather your rods and cones be fatigued while you're sleeping or while you're crossing the street? Which sounds safer to you? Yeah, it's while you're sleeping, right, Jakia? It's not while you're crossing the street. You don't want to get halfway across the street and go, oh man, I ran out of neurotransmitter. I guess I'll just wait. Because I'm sure all the cars will wait too. And then what happens if one of those people driving ran out of neurotransmitter? Like, oh, what was that? So, it's kind of a good thing. Because, again, we are diurnal animals, so we're going to roll with that. This is sort of a complicated concept, but I want to make sure I get this across to you. And that's the concept of a receptive field. Okay? You have rods and cones. Each one of those rods and cones is responsible for a small piece of the world, right? That's its receptive field. So you've got a rod and it's located here. We can make it a cone. If you think about the visual field, it's really sort of hemispherical. And let's assume you've got really great sort of peripheral vision. You know, you could see like 180 degrees, right? Okay? And somewhere out in this, you know, because you've got these other sort of right? So that kind of looks like a hemisphere, right? It's kind of a globe, okay? And you've got all of these rods and cones on your retina, and they're responsible for all of these little spaces, wherever they are, right? Okay, and there's going to be enough of them to kind of overlap and fill in the whole world, right? That whole visual world you can see. So that's going to be awesome. Okay. So each cell's receptive field is going to be that little area there for which it's responsible. That's pretty cool, right? Something happens over here, this guy doesn't get excited about that. Why? Because there's some other cell that has a receptive field over here that's going to take care of that for you, okay? This guy gets excited when light hits this part of the retina from that part of the world, right? And where is that part of the world? Well, it's wherever you're looking, right? Because your head's constantly moving, your eyes are constantly moving. We want to think about that visual field not as absolute coordinates out in the world, but as relative coordinates related to your, your visual fields. Okay? Does that make sense to everybody? Good. We can talk about eye movements. Maybe, well, we should talk about eye movements. Your eyes are always moving. There are a couple kinds of movements you can make. One are what we call vergence movements, right? So that's if you're like following something. You can do this with your finger if you want. You know, your eyes will kind of come in and out to help you get focused. Saccadic movements, these are, you're looking over on one side of the world and all of a sudden you want to move your eyes somewhere else, right? These are like rapid eye movements from one location to another. So if there's like a picture, you might, make these rapid eye movements kind of all over the place, right? The other movements are what we call smooth pursuit. This is when you're like watching someone walk or you're trying to like follow something. These are impossible unless you're actually tracking an object. And if you don't believe me, you can try it. Try to imagine, like a bubble floating across the room. And you'll do it and your eyes will jump. You have to start over. And then your eyes will jump again. And so it's totally impossible to do smooth pursuit unless you're actually following an object, right? You can do it with your finger. But if you imagine that finger moving, your eyes are just going to jump. Give it a try. That should be another exercise we add. Yeah. So you can do that. Just write that down and get this started. Yeah. What else do you guys need to know about eye movements? I don't know. I think that's probably pretty good. Not only does your fovea have only cones, the way the cones are wired up to your bipolar cells will actually give you much better what we call visual acuity, right? Okay? This is again why when you're reading, when you're looking at something, you put it on your fovea. You don't put it out in the periphery. And again, this bothers me, so I'm going to fix it. These should all be rods because we're in the periphery, right? You don't have cones in your periphery, so let's fix that. In the periphery, you might have 5 or 10 photoreceptors headed down to a handful of bipolar cells going to one ganglion cell, right? So that one ganglion cell that's going to fire that beautiful action potential is pulling information from like 10 photoreceptors, right? And those 10 photoreceptors let's just draw them here. 9's a good number because that's a square number. So there's 9. 9 photo. Look at that. Okay? So you're not getting 9 individual blocks there. You're just getting one big block, right? So if you want to think about this, this is kind of like 8-bit Mario. right? I don't know if you guys have played like old school video games, right? And everybody's like square, and you punch somebody with a square fist, right? So that's how that works. This guy, we've got one cone, one bipolar cell, one ganglion cell. So it's going to be like this. And then next to it is going to be another, but they're all going to be different colors, right? Because they're all different photoreceptors. These aren't the same photoreceptors, so what we're getting is a much better image of what's going on in the world, right? So this is sort of like, 4K HD or something, right? You're going to play your newest video game and it's going to be in HD, right? So you're going to get those smooth edges. So you have much better acuity in the fovea than you do in the periphery. And that's because, again, of the wiring dynamics here, right? That wiring diagram makes all the difference. Optic nerves. What else do you need to know about this? They're leaving out of the retina through that blind spot, through that optic disc. We're going to collect all of that to make the optic nerve. The optic nerve then travels to a guy called the dorsal lateral geniculate nucleus. The LGN, typically the DLGN, right? Because the MLGN is something else. This guy's in a structure called the thalamus. Okay. We could talk all day long about the LGN and all of the cool features there. It's got a 6-layered structure plus a bunch of other layers that we figured out later actually do something. We'll talk about that briefly. We can talk about the developmental mechanics of this. I mean, it really is a fascinating thing. But I'll tell you what's important. Oh, we should also talk about the partial decussation. How many of you remember that left-right cross in the nervous system, right? Same thing happens with vision, except it's not complete. So the information that comes from your left eye does not entirely go to your right brain, right hemisphere. And the information from your left eye doesn't, or the right eye doesn't all go over to your left hemisphere. Part of it does. What's more important is actually the visual field, right? So things that are in the left visual field end up on the right side of your brain. Things in the right visual field end up in the left side. Now, how many of you can close one eye at a time? You can try it if it's an easy thing to, yeah, there you go. Great job. If you close one eye, what you notice is you can see some stuff across midline, right? So you can see some things like on the other side, right? That information is already where it belongs, right? Because it's already coming from the other side. So that doesn't have to cross over, okay? So not all of the information crosses over. So if you look at the information here, goes there, this information already on that side, right? That's why we call it partial, and decussation just means crossover. This guy's known as the optic chiasm. How many of you are in, you don't have to answer this, a fraternity, a sorority, or in a group of nerds who know the Greek alphabet? So chi is the Greek letter X, right? So it makes an X. It crosses over right there. All right. Once we get into the LGN, cool stuff happens. We'll talk about that. From the LGN, we're going to go to an area called the primary visual cortex, known as V1. Okay. From there, we actually go to a variety of other areas, not just V2. There's V3, V4, V8, MT, MST, TEO, all kinds of things that you don't need to know right now, but you will in about 5.5 minutes. I'll give you 6. You'll have to know it later. Again, here's that visual pathway, again, demonstrating that partial decussation. But also demonstrating, because this guy's the thalamus at LGN, demonstrating the path from the LGN back to your striate cortex, which is also known as your primary visual cortex. It's also known as V1. It's all the same thing. Why do we call it the striate cortex? If you look at it, it looks like it's striped. We'll see that in a minute. All right. Let's talk briefly about your LGN, your lateral geniculate nucleus. Why do they call it the, what does geniculate mean? Anybody know? Genu is, the knee, right? So it's, in the human, it's bent. In other species, it's not. But in the human, it's actually, and I think most of you are humans, it's going to be bent like this, okay? Which some people actually say looks like a Napoleon hat, like Napoleon's hat, right? So you can kind of see that. So there you go. There are, they will tell you 6 layers of neurons. That's not true. There are 6 layers in what we call the magnocellular and parvocellular layers. There are other layers that were discovered later, and people realized they were important, and so they, but they didn't change the numbering structure. So it's not important. Some of these layers get information from the opposite eye. Some of them get layers from the same eye, right? It's the eye on the same side. Magnocellular, magno means large, parvo means small. That's related to the receptive field size of the ganglion cells that are coming in. Okay, we'll talk about that. Coniocellular, that means dust. Originally, people thought these coniocellular layers were not that important. They had like small numbers of cells in there. He said, "Oh, that's just dust. It's not important." In fact, it actually might be processing color vision. It's actually kind of an important layer. All right, there you go. That's your DLGN. Notice how it's bent, right? Okay. You've got your parvocellular layers and your magnocellular layers. You have more parvocellular layers than you do magnocellular layers, right? And again, these are going to be smaller receptive fields, which means you're going to have higher visual acuity. Okay. I'm thinking about when's going to be the best time to let you watch a movie, because I have a great movie for you to watch. You might be getting there. We should talk about these 2 guys, David Hubel and Torsten Wiesel. These guys won a Nobel Prize. They won a Nobel Prize by figuring out how visual cortex responds to specific features. In this case, you were thinking about contours, so like shapes, whatever those are, right? What's cool about this is, remember those little receptive fields that we have? And they're typically circular. Okay. What happens if this guy, if those guys are activated?
They activate the nearby other neurons.
They will. No, you're absolutely right about that. So the other guys will get a little excited. And we want to take this information, not at the retina. At the LGN, we'll do a little bit, but by the time we get to cortex, we want to take a look at this and not just look at this as, 5 or 6 independent dots, but we want to put it together into a shape, right? So that we can start to know how these receptive fields are connected. And if adjacent ones are activated, we need to know if all of that activation goes together, right? Can you imagine if you were viewing the world and it was all independent little dots, and you didn't know if one dot next to the other was actually connected? Can you imagine how difficult it would be to walk out the door? It'd be impossible, right? You'd have to kind of crawl on hands and knees over there and feel for the door frame to tell if, the posts and the jamb were connected, right? Thankfully, we don't have to do that. And that's because David Hubel and Torsten Wiesel made certain of it. No, they described how this happens, right? But your visual cortex will take care of this for you. Let's see. I do think now might be a, well, let's go just a little bit more and then we'll watch a video. All right. So we've thought about how this works. Again, we've got the 6 layers of cortex. There are these cytochrome oxidase blobs. I'm not going to expect you to know much about that. So this is sort of the structure of cortex. Remember I said you've got some layers that have sublayers. So that's all coming back here on your primary visual cortex. Don't worry about this thick stripe to thin stripe business. That's not things I'm going to ask you about. Once we get above or beyond area V1, we go into some of these specialized areas. We will talk about those here in just a few moments. There's a general approach that we think about as the dorsal and the ventral stream, right? And this is sort of a what versus where pathway. Now, I think it's interesting that they're structured that way. I don't know that that's going to provide you a lot of information at the moment. We'll think about the what and where pathways we need to, right? Because there's some things that are going to be telling us the identification of what we're seeing, and other things will tell us the location of that, which is important, right? How many of you love cars? Okay, cars are fine, right? How many of you made it to campus using a car? And not just like immediately. You may live on campus without a car, but at some point you use the car to get here, right? So you probably have been in a car at some point in your life. That's phenomenal. What you don't like are cars coming at you while you're on the road, right? Like down the wrong lane. So you want to be certain that that what it is where it should be, right? And so they're two separate pathways. So there you go. Not a big deal. Nothing to worry about. I do want to talk about this because this is going to relate to the video that we're going to watch. Now these are ganglion cells. These are not cells in the cortex. These are not cells in the thalamus. We've zoomed back to the retina, right? Remember those retinal cells. These guys are firing an action potential. This is the shape of their receptive field. It's got two components, a center and a surround. Okay? Not a big deal. What are we going to do with the center and the surround? We can do a couple things. Basically, if light hits on the center of this cell, it's going to get activated and fire more action potentials. If light falls in the surrounding area of the cell, it'll get inhibited, and notice there are fewer action potentials. That's what we would call an on, or on-center ganglion cell. We can flip this and have an off-center. That's fine. If we activate the center of it with light, we don't have action potentials. If we hit the periphery, we do get action potentials. Why is this important to you? Think about how we might wire these ganglion cells so that we can get the type of... We want to be able to take these circles, I like to call them donuts, and we want to eventually create boxes of donuts. And this is what we'll see in just a few minutes, where we can line up these activated, these centers to get an area of activation, and then we can get what we call inhibitory flanks. And that'll make a lot of sense once we watch this video. All right. It's interesting. There's a video with it's from PBS apparently, and a cat with an eye patch. See that over here? Who knows if they know what they're talking about. All right. This was a set of experiments by David Hubel and Torsten Wiesel. They did most of these experiments originally back in the mid to late 60s. Continued on for a while. Did a variety of other things. Then they eventually got a Nobel Prize in the 80s for their work in medicine or physiology. They are recording, and you'll hear this, so I have to turn the volume. They are recording action potentials from cells in the cat visual cortex. You will not see the cat. Okay, I want to kind of put that out there for you, right? Sometimes that can freak people out. Okay, they've got an electrode. Remember those electrodes we talked about? They've inserted that into the cat cortex, and they'll move around until they find a cell that's active, and they'll find a cell. All you will see really is the screen that the cat is looking at, right? But while you're thinking about this screen, think about that visual field, right? And map it onto this screen, okay? What these guys will do is they'll use a spot of light or a bar of light. They will find the edges of the cell, okay? When they find where it's active, they'll, make marks on the, because it's like a big roll of paper. So they'll make marks on there where it's active. They'll make marks where it's inhibitory, right? So what they're showing you here, there are a couple different types of cells in the cortex. Some are what we call simple cells. Simple cells have an excitatory region, typically in the middle, and they have inhibitory flanks. So if you put light in the middle, the cell gets excited. You'll hear that. There are like these pops and cracks that you'll hear. When they put the light on the inhibitory flanks, you'll notice there's a decrease, okay? When they move to the complex cell, there is not an inhibitory or excitatory subregion set up. It's all going to be excitatory for the entire size of that receptive field, okay? Casey, this video changed my life. I hope it changes yours. No, when I saw this video, I thought, wow, that brain stuff's pretty cool. Maybe I should learn about that. It was a big mistake. No. Just kidding. All right, are you guys ready? hope that made some change in your life. Probably did. Casey, are you going to abandon your plans and
At this point,
you should?
but it was really cool.
It was still really cool, wasn't it? To think that, I mean, one, it's fascinating, right? Because you can actually hear action potentials, right? We've talked about that simply as, kind of that. But to be able to convert that into something that you can hear and you can measure and you can actually experience is actually pretty fascinating. All right. So, who wants to talk about color vision? I'm going to underline this in red for you. Let's see. So, trichromatic vision. There are different types of color deficits. There are folks who only have one sort of cone that doesn't work or is missing, 2 or 3. So that's a problem. Some folks are completely achromatic, so they can't see any color at all. They only see things kind of in grayscale. That's pretty rare. Even protanopia, which is the most common, is fairly rare, right? I mean, we're not talking about like half the people have color deficits. In fact, is it maybe 2 or 3% of males are colorblind? I mean, that's females typically aren't colorblind, based on the way things work. I don't know why this is down here. So, we want to think about color vision. Why is color vision important to us? There are a number of reasons why color vision might be important to us. One of the most common theories is that it was for fruit detection, right? How many of you love fruit? How many of you love rotten fruit? Yeah, you know how you can tell if fruit is rotten? Look at the color. Right? You can typically look at the outside of the fruit and go, that's ripe or it's not ripe, or it's overripe, right? You can kind of tell that. So that's one possible theory. The other is maybe related to fertility. I know you don't really think about like color vision and fertility. There are certain species that will turn parts of their body bright red when they're fertile. Right? And so if you weren't able to see color, you wouldn't recognize that, hey, something's bright red, maybe that's time for us to mate. So that's a possibility as well. Not all species have color vision. Not all species have trichromatic color vision. That's really reserved to primates, right? Once you get outside of primates, you start having different types of color vision. They're not quite as advanced. You do have 3 different types of photoreceptors, cones, that absorb different wavelengths of light. So here are your rods. You've got what we would think about your blue cone, but really that's going to be short wavelength. You've got a green cone, which is kind of a medium wavelength, and then red cones, which are long wavelength. These 3 cone types absorb wavelengths of light at different types of wavelengths. Right, and they will send those to different bipolar cells and get that information into your brain. The most common type of color deficit is what we refer to as red-green color blindness, right? And this typically occurs when there's a problem with your red or green cones. And what will happen is these 2 wavelengths of light, these 2 peaks, will actually get closer together. Okay? And if they get closer together, you have a bigger overlap, an area of confusion, right? And you're more likely to think something that is red or perceive something as red when in fact it's what most people perceive as green, because the overlap on those cones are much closer. Those cone absorptions. We call it absorption spectra, but you don't need to worry about that too much. All right. This is how you can test for color vision. Has anybody taken a color vision test before? Yeah, they're fairly common, right? So you can take one of these. You should be able to see these numbers. And if you don't, you should probably get your color vision checked at your doctor. So I would say most everyone can definitely see that that's the number 1, right? If not, then you've got a problem with all your cone types. Going back up here, you hopefully can see that that's a number 5. There are a couple of you who might not be able to see that that's a 29, but most of you should, right? Again, color blindness is not that common. You know, it's a small percentage of folks. If I were to ask you to tell me some of the basic colors, you would probably list red, green, blue, and yellow, right? If we think about those 4 colors, there's not really a way to divide those down into something else. If I said orange, you might think of that as some combination of red and yellow, right? But there's no way to think of blue as a combination. Well, this doesn't work, right? Like, you can't make red out of these other colors. Conceptually, it doesn't work very well. And it doesn't really match up to the cone types that we have. Except this guy yellow here. That's kind of a crazy one. So, on top of that trichromatic color vision, we also have what's called opponent color processing. So we will wire up red and green, but we will also wire up blue and yellow. And how we get the yellow is that's made up of a red and a green. So there you go. You don't have to worry about that. I'm not going to really ask you. This is sort of interesting. How many of you love these negative after image? Anybody seen one of these before? You can stare at this apple for somewhere 40, 50 minutes and don't blink. And then if you look at the blank sort of area there, you should see something fascinating. It says, "Paul is not dead." It was a joke. Some of you understood. Great job, Casey, for laughing at that. I appreciate it. So how does this work, right? So this is an after image. Hopefully when you looked over here, you saw like sort of a red apple and then some kind of green stem, right? Did most of you get it? Did anybody try it and actually get that? I've seen things like this before, right? So how does this work? Well, it works by like saturating things, right? So if you continue to stare at this area, all of those cones that are sensitive to that wavelength of light are going to become fatigued, right? And they're going to drop out. And then when you look over on somewhere else, whatever photoreceptors are there, especially those opponent color ones, are still going to be relatively active, right? Even if it's just accidental baseline activation. But because you fatigued sort of all of the green, that red, even though it's not getting activated, seems like it's activated, right? So this is confusing, right? It can really trick you. This is why you should never grade on a curve, right? Why'd you make that face? Why wouldn't you say that in here?
They might want to be graded on the curve.
Well, you never should. Because what could happen is instead of all of you doing really well, like one of you could do okay, and then the rest of you could just be horrible. And then I think, that guy's a genius. But it's just the rest of you were idiots. Right? That's what's happening here. It's not that the red is getting activated. They're like, wow, that area is really red. It's just that all the green is not doing anything. So it makes it appear more red, right? So that's the trick. Convince all of your peers to really be horrible, and then you'll look better by comparison, even if you don't improve yourself. That was good, wasn't it? Don't worry too much about this. I could ask you a lot of questions about that, but I'm not going to. Color processing, most of that's going to be coniocellular, some of it parvocellular, not a big deal. All right, let's think about other stuff, right? Let's kind of zoom out a little bit and think about what we call the extrastriate cortex. So we are beyond V1, right? And we're thinking about higher levels of processing. So we're taking all that, those little dots, we took all of those dots and donuts, we put them into boxes of donuts. We need to take those boxes of donuts, those bars, right? We've connected those contours, and we need to turn them into things, right? Like doors and chairs and cars and pants. I don't know, right? I'm going to think about things you all know about. Short list. So, let's think about color for a moment. How many of you love V8? And not that beverage that tastes like cold tomato soup, because that's nasty. And it's salty. It is so salty, right? Has anybody ever tried to drink V8? Yeah, waste of money. You're better off just go suck on a tomato, really. It tastes better, and it's probably better for you, because it's not going to give you high blood pressure. I just feel like all the salt, I don't know. So these folks did a study with an fMRI. They put folks in a machine, right? They show you some pictures. Some of them they show you are achromatic. Some of them have color. You can do some subtraction, right? Between then, you can see which brain regions are active when they're viewing color images. And they called that area V8. So that's great, right? That's phenomenal. But we want to see what's the sort of opposite study of that. Well, the opposite thing you can do is go around and cut out people's V8, right, and see if they can still see color. Now, there aren't a lot of people who are going to let you do that, but you can find people who've actually had damage to area V8, and then you can give them a color vision test, right? And so these folks did this, 10 or 12 years ago. They actually found 92 people, right, who had damage to V8, and they found that they all were achromatic. Now, you need to confirm that this is not a retinal issue, right? So you're also going to have to check to make sure that they have all three cone types and that they all function. So you need to do that to verify that it's actually brain damage causing that and not something else. And here you go. Here's area V8, just to sort of show you where that's located. So that's color. What about form? Remember, in V1, we just had these little bars, right? Just these little bars of light, whatever. We want to take those bars and we want to put them together to make shapes. Okay? Shapes help us, right? So we know what something is. Okay? That's going to happen in an area called V2. Not a big deal. And we're not going to worry too much about spatial frequency. But V2 is going to help you with this, what we call spatial vision. So taking these individual pieces and kind of putting that together into a form. If we think about filtering out spatial information, right? Okay. We can look at these two images. In case you don't know who this is Abraham Lincoln. That guy was important. I don't know if you guys know this. I mean, without him, we wouldn't have pennies, right? I think he did something a little more important than that, than just like loaned his head to be on the penny. But you can ask the folks down the hall about that, because that's the history department. They'll tell you all about the great Abraham Lincoln. Did you read that book, Team of Rivals? Can I put in a plug for something? You should read this book called Team of Rivals. Like, so I always thought like Lincoln, like he's like this super honest, cool guy, right? Honest Abe, right? And he's like chopped down a tree by looking at it with honesty. I don't know, right? Like that's the legend. And he is super cool. But there's this book, and so here's the reason I'm plugging this, because I know you guys won't go to this, but you should. There's a book called Team of Rivals, written by Doris Kearns Goodwin. And Doris Kearns Goodwin is probably one of the, she's written a ton of other books, but she's probably one of the best historians right now, like out there doing things. She wrote a book called No Ordinary Time about the FDR administration. She's written some. She actually was a staffer for Lyndon Johnson, and she actually attended some of his toilet meetings. Is that what you call those? Like he would apparently have staff meetings while he was on the can. It's like a true Lyndon Johnson story, right? You guys, just in case you want to know about Lyndon Johnson. Anyway, she wrote this book, Team Arrivals. It was a phenomenal book. She wrote a bunch of other books. She's written books about awesome things, right? And folks, the book Team Arrivals is great. It actually tells you about some of the political genius of Abraham Lincoln, right? You really just think like he just won everything because he was honest. Man, the guy was brilliant. And he would like pick people and convince them to do things, and the way that he would do that, like in anybody else, you would call him an evil genius, but he was doing good things. So I guess he's a benevolent genius, right? But that just doesn't have the same ring to it. You should read this book. It's phenomenal. Like he just picked these people who hated each other, but managed to get them to work together to accomplish things. It was great. Doris Kearns Goodwin is coming to town in October, I believe. Yeah, so that's going to be pretty awesome. I'm kind of excited about that.
It supposedly has really good reviews too.
Team Arrivals? Yeah, so there was this guy named Barack Obama who said this was one of his favorite books. I don't know if you're familiar with him. He made a book list, and this one was on it, and I read it. He actually read this while he was like running for president, and so that kind of, you know, informed his political sort of maneuverings. That still sounds devious. His political decisions in terms of who he would put in his cabinet. That's a total side story, although I think, you know, side stories about Abraham Lincoln are probably worthwhile. But you should try to go to see Doris Kearns Goodwin when she comes to town, because these authors, when they come in, and I don't know how much it'll cost, but I think students could go for free. It's part of the martial artist series, so you guys might get free tickets. But typically when they come in, they're not well attended, and that's really sad. It is sad because, you know, everybody will go see, like, I don't know, they bring in good people, so I really even hate to say, like, you know, everybody's gonna go see Steve Martin when he's here, but I mean, there's some value to seeing Steve Martin as well. We should see Doris Kearns Goodwin. Anybody a baseball fan? She's supposed to be working on a project with Bud Selig. Yeah, I know. I'm pretty excited about that. That's the only reason I'm going is to ask her about it. To see if, it's like, how far along is your baseball history, Bud? Did you see the documentary by Ken Burns?
Mm-hmm.
She's featured it.
Oh, okay.
Yeah, she talks about when the Brooklyn Dodgers left and like how sad her
Okay,
family
yeah,
was.
okay.
You know who she is now.
I knew that name sounded familiar.
So, these two images have the same amount of what we would call low frequency information, right? But we've added some high frequency information or filtered it from this image, right? And so you don't see that, and it makes it look completely different. And actually, Riley, for you and Alex, they might actually look very similar that far away. Because when you get that far away, the information sort of, like if you were to get farther and farther away, the information sort of filters out, right? But if you're close to it, you can see that it's completely different. So that's fun. Who wants to get up and go to the back of the class and see this? I should make everybody do that. Here's what I should do. Like, I should say it's attendance time, and if you're not in the back row, I'm not counting you. That would be mean, wouldn't it? I won't do that. As much as I want to. What are some other things in extra striate cortex? There are a couple of areas called TEO and TE. These are actually for recognition of visual patterns and objects. So if you lesion those brain regions, animals can no longer recognize patterns that they may have known before, right? You can associate a certain pattern with a treat or a shock, for example. Get rid of one of these brain regions, and guess what? They have no idea which one to select. Don't worry too much about this one. We should talk about some of this stuff, though. These are some interesting areas, interesting things we need to talk about. Anytime we're talking about an agnosia, then we're talking about something that you can't see, right? So there are different kinds. Here's one for faces, which is kind of cool. There's actually this area called the fusiform face area, the FFA, right? This is pretty cool. How many of you think it's important that you can recognize faces? Yeah, it is. If you don't believe me, try to convince this to your significant other. The next time you like go into the wrong home, trust me, the excuse, my fusiform face area wasn't working, will not get you out of trouble. I guarantee you. So don't try that. Okay? So you're just trying to arm you with some knowledge here. So the fusiform face area, it is really interesting that it works for facial recognition. It also seems to work for other types of things. Like, how many of you are, anybody an ornithologist? Ask about ornithologist. Nobody's an ornithologist. Nobody's a bird watcher or a birder, which is the correct term, right? Anybody collect something? Something you want to share?
Yeah, I collect the little pop figurines.
Got it. That's perfect. And they make different versions of those. They do. And for someone who's not familiar with those, they may go, that's just a big square-headed plastic thing, right? Someone who does collect them may recognize that that is the, I don't know, whatever special edition version of the Incredible Hulk with the purple pants instead of the gray pants. And that, I don't know, I don't even know if that exists. I'm making this up, so don't go out looking for it. It could exist. Very well could. And that might be something that means something to you, right? Your fusiform face area is going to get excited when you see those because you've spent a lot of time like with these fine discriminations, right? So how many of you have ever seen like an elephant? Yeah, and how many, you've seen multiple elephants, right, Zane? Huh?
Yes.
Yeah, sure, multiple elephants. Now, if I were to line those elephants up to you and a bunch of other elephants in an elephant lineup, and I need you to pick which one you've seen, you got no idea. Just a bunch of elephants, right? Okay, and if I lined a bunch of us up in front of an elephant, the elephant might have the same difficulty, right? Like picking out our faces. Because if you look at everybody's face, and I can see most everyone's face, they all look the same to me, right? You've all got noses, you got mouths, you got ears and eyes, you got some hair in a couple places. There's not a lot of variety, right? And if I'm not an expert at facial recognition, which believe it or not I am, and so are you, like you're all experts at this. If I was not an expert at this, then I would just, I don't know how I would tell you apart, right? Because all faces contain very similar things. Now we don't think about this because we have spent our entire lifetimes looking at different faces and knowing that those faces are different, they belong to different people. But when you look at people's faces, they all have the same elements, right? They all contain the same things. And they're very, very close to one another in reality when you look at this. They're much closer. I could randomly pick any two faces. They're going to be much closer to each other than if I were to randomly select a rock and a piece of fruit, right? So if I'm comparing a rock and a banana, or I'm comparing any two people, those two people's faces are going to be much more similar than that rock and banana, right? And so it's really important that we have an area that we can identify each other, right? And that's going to be our fusiform face area. So we're going to look each other in the face, and we're going to go, I know who that is, or I don't know who it is, but I know it's somebody different, right? You don't think about that, like how similar faces are, but they are. Who wants to hear a story about, I should tell you about chimpanzees. Yeah, and their butts. So this is a two-part story. One, there was a study a few years ago where they did some eye tracking studies on chimpanzees. So they had a male chimpanzee, and they put him in this, you just like sit down. You like track his eyes, where he's looking, right? And they had a video of a female chimpanzee like walking across the screen. And the whole time they're tracking his eyes, it just goes face butt, face butt, face butt, face butt. The whole time, that's where his eyes are going. Face butt, face butt, face butt, face butt. The whole time she's walking across the screen, right? And so you're thinking, okay, well that's a little weird. But what's really interesting about a chimpanzee's butt is they're all unique, and they have unique butt wrinkles. See? And so while we're face experts, they're butt experts. And so they will look at each other's butts to identify them, right? And so based on the pattern of the wrinkles, they'll know who it is. So they can look at their butt and go, it's my Uncle Charlie. They can look at the butt and then go, that's Sally, and they know. So they probably, I assume, they have what I like to call an FBA, a fusiform butt area. They probably use some like fancy word for butt, right? But I would just make it butt. Or I would maybe probably call it the FAA. But that's me. So there you go. There's a chimpanzee butt story you didn't think you were going to get today. But that's how they identify each other. There are other animals that can identify faces. One of those are paper wasps. This particular species of wasps, they actually have facial identification. Now, you think, why in the world would an insect be able to look at another insect and know it by its face? The key sort of common element here is they're all social. They're all social species, right? So in a social species, it's important to know who you are, who somebody else is, and the interactions you've had with other people, right? If we were not a social species, if we were solitary and we just like randomly come up on another member of our species once every 6 months, who cares who it is? It doesn't really matter, right? But when you're living amongst all of these people, it's really important to know who each person is, what they've done to you, what you've done to them, and how you can avoid them if you owe them money. Right? Those are important things. That's exciting, right? Fusiform butt area. I don't know. So, you know what I should have done? I should have had a whole slide of chimpanzee butts with different wrinkle patterns. And then see if you could identify who they are. What are you guys going to tell your parents when they ask you, like, because I know some of you like call your moms every day after class, right? Anybody in that, you don't have to admit to it. I know there's like someone who calls their mom every day after class, right? That happens. What are you going to tell her? You know, like, we learned about chimpanzee butts today. Do you want to hear about it? They have different wrinkle patterns. That's some really good vision, because I'm thinking about a chimpanzee's butt now, right? I'm thinking, like, how close do I have to be to see those wrinkles? Right? I'm thinking that I'd like to be able to identify you from a distance if I've got to do it, you know, through your butt. So I'm just like, the farther away I can be and know who you are, the better. Oh, I should tell you about the extra striate body area. This is part of your brain that gets excited about body parts. Just like random body parts. If there's an arm on the ground somewhere, you know, that, no, seriously. There's also the perihippocampal place area. We can talk about that later. So here you go. Again, this is the fusiform face area. It gets really excited by faces. Also, it gets a little bit excited by headless bodies. I don't know how many of those you run across. I guess it depends on your line of work. The extra striate body area gets super excited about headless bodies, and then just like random body parts as well. Okay. We'll talk about places later. Let's see. Oh, so here's what's interesting. Are there developmental differences in how we respond to faces, right? And the answer is yeah. So here's a child's brain, here's an adult's brain. They've been sort of scaled to match for size because an 8-year-old's brain's a little smaller than an adult, right? It's not like full adult size. Here's what we would think of as the fusiform face area in an 8-year-old, right? And then here's what we would think of the fusiform face area of an adult. Now, if you would, let's think about all of ourselves when we were 8 years old, and how many faces had we seen compared to how many faces you've seen a few decades later, right? You've seen a lot more faces. You have to keep track of more people, right? So there you go. So it does have some change. It can change. That's cool, right? Do we want to talk about retinal disparity? Maybe we do. You have binocular vision. Both of your eyes point forward. There's a lot of overlap, right? That's great for depth perception. So you can kind of have these two slightly shifted views of the world. We call that stereoscopic vision. Not a big deal. If we want to know how far something is away from us, there's some things that we can do. If it's bigger on the retina, then it's probably closer, right? That only works if you're comparing things of a similar size. If an elephant takes up more space on your retina than a mouse, that may not tell you much. But if the mouse takes up more space than the retina, you know the mouse is like clawing your eyes out. because it's right there. We already talked about this. Striate cortex. We talked about all of this in that orientation selectivity business, right? You saw that video. That was super awesome. Optic flow is kind of cool. How many of you have ever moved? Like through space and time? Happens all the time, right? Things flow by your face, right? By your head. That's kind of cool. Think about walking down a corridor or an alley. Right? So there are some folks who cannot perceive movement. That's bad. You know why that's bad? Not only can nothing move around you, but you shouldn't move either. Okay? So that's not bad. They get like static images, right? So that's not cool. And that's going to be damage to an area called MST, or V5. It's the same place. And that's going to take place out here in the temporal lobe. Don't worry about that. This is a chart telling you sort of what all the visual areas do. You can look over that if you want. Any questions about